So thank you all to everyone who's come today to the talk with Professor Tonio Enriquez. Very interesting work that I know that he's going to present today, but I just wanted to give a very brief introduction. Going way back, you had a really great interest at your PhD. You were in Spain, University of Zaragoza, known as UNIZAR, under Julia Montoya and Manuel José López Pérez, and investigating mitochondrial DNA biogenesis. So there's some real hardcore mitochondrial biology that you were investigating, and also later on the more structural aspects and organization of the key elements of the OX-PHOS pathway. Some really hardcore mitochondrial biology, but I think it's really evolved over your career, especially when you return back from Caltech. You'd already looked at the pathogenicity of tRNA mutations in mitochondria and beginning to get interested in disease. But I think then obviously you had the opportunity to form your own research group. I think that was in 98, 99, and more recently affiliated or now moved over to the Spanish National Centre for Cardiovascular Research, where fundamental interest in mitochondria and pathogenicity and metabolism in involvement in physiology. And I know there's many areas that you have evolved and made important contributions towards, particularly I think there was a Nature paper in 2016 where it's showing that natural variants of mitochondria had profound effects and aspects of physiology and also metabolism and longevity. I think they're really interesting. And then more into metabolic adaptation with relevance to immunology, and also in context of cardiovascular disease. There's been very important works that you've contributed towards. And some of you here today, I hope you might recognize Tonio. He did a sabbatical, which was then at the Dunn Nutritional division, which is now known as MBU, the MRC MBU Centre. So hope there's going to be someone of you to ask questions. And obviously more recent work and co-authors even with James Rudd and others, but also is interested in metabolism, in immunology, in the context of obesity and macrophages there. So it's been some wonderful work, and I know that you're going to be talking to us about the work more recently in the Cell paper, where you formed a very important collaboration with an immunologist, Andreas Hidalgo, if I've pronounced that correctly, and we look very much forward to the talk today. Thank you.
Okay, thank you, Jan, for the introduction, and thank you for the invitation. For me, Cambridge is a special site because I spend almost a year with my family and having a very, very good time. So before I started, I really wanted to really indicate that this work was initiated by Professor Andrés Hidalgo at our institution, and led with a PhD student at that time that was José Ángel Nicolás Ávila, because they are really studying macrophages in the heart, and they make a discovery that led us to call us that we were on the other side of the building. And then Ana Victoria Lechuga, that was a PhD student in my lab, just took responsibility of this collaboration. And now she's in England, but it's not in Cambridge, she's in Oxford. And then they call us because our expertise in mitochondria, and we together try to really correlate these three phenomena: cardio-functional homeostasis, resident macrophages in the heart, and the mitochondrial function of the heart. Okay, but it's not only these two groups. Actually, this was an effort of the whole institute that you have here in the picture. The paper was already published at the end of the last year, so you can follow it if you need it. But there is a number of PIs of our center. You see that they needed to help us to understand and to develop all this work, and also a number of units that are represented from the bioinformatics to in vivo image and transgenic and so on. So there is really a choral work that came from the whole institute. I think that this is a scenic paper rather than a group paper. So you know that I, as a mitochondriologist, I used to put this slide as to make it clear to the the cardiologists that there is the principal and major reason because the whole system of cardiovascular system was developed is just to deliver oxygen to all and every of our cells. There are many other functions that are coming on top of this, but this is just critical in terms of moving the origin of this system, but also in timing. This is the only function that it cannot stop at all during the life of an individual. And the reason because it has to circulate oxygen and take back carbon dioxide is because the oxygen has to go mainly, not exclusively, but mainly to this organ, the mitochondria. So, and what happened in differentiated organisms, mitochondria is present in every eukaryote cell, or some exceptional situation, they lost it, but they were originally in all of them. But there is, as you see, many cells on the body that are packed with mitochondria, like this is brown fat, for example. But the cell that is more really, one of the cells that is more really dependent on mitochondria is the cardiomyocyte. Mitochondria are really labeled here in red. You almost don't distinguish when one mitochondria ended and when the other started because they are pretty close and very well organized. And it occupied the 40% of the volume of a cardiomyocyte. So this is really from the point of view of making this type of cell, this is really overwhelming, the work that this cell has to dedicate to maintain these organelles, not only these organelles, actually together with also packet structures like these sarcomeres and all the compartments that are need to maintain the flux of electrons to allow this cell to function. So this is a tremendous, overwhelming structural organization of this. And this structure has to really be replaced, maintained, and keep in quality. And particularly in the mitochondria, the way where the oxygen goes is to this guy. This is the complex 4 that is actually trying to capture electrons to form water, electrons that has been obtained by the digestion of different metabolic process, and then they are using them, as you all know, to generate forces to transport protons across the membrane, to generate also the electrochemical gradient to allow the complex 5 to generate ATP. But also, there are components here that are needed to be recycled for many other aspects of the metabolism different than the ATP synthesis. But instead doing that, as you see in here, that the oxygen is actually burning the organic compounds, this has to be done at room temperature in a well-organized form. And sometimes the electrons can go out to form toxic compounds like oxygen radical superoxide and peroxide. So to maintain this function control, there is a quite complex quality control capacity at different levels, at the level of the proteins with chaperones and proteases, preventing the ROS to go to damage cells or other mitochondrial components. They have several antioxidant systems. When the damage is done, they have capacity to repair DNA or lipids and also to modulate the function of the mitochondria. But if the organelle is damaged as an organelle, it can really try to be eliminated by a specific degradation of the whole organelle by mitophagy. Because it's this fail in all aspects, then you can induce cell death through apoptosis, and then the quality control is critical. The figure of the life cycle of the mitochondria that was developed by Orient City High group, that is a continuous fusion-fission phenomenon trying to keep healthy this mitochondria. and they are monitored by the membrane potential. And when the membrane potential goes down, there is really a decision point in which you recover it by the low-level quality control, or you send it for degradation to prevent the apoptosis. So the question is that when you are a neonatal cardiomyocyte, you have a quite high number of mitochondria, but the mitochondria can't really move and organize in the cytoplasm. But when you go to an adult cardiomyocyte, the organization is extremely complex. The mobility of the mitochondria is very compromised. And then how you can really maintain the quality of mitochondria in every part of the cell functional. And the important question also is that you have to do that while you are maintaining the function. You cannot; these cells cannot rest. So beating must be maintained and go all your life, and it cannot stop even minutes. The number of beats or heart rate is enormous, and even is more in the mouse. And the number of beats per life is enormous. So you have a very complex cell. You have to really maintain the quality of these guys that produce toxic effects if they are not good quality, and you cannot stop the function that you have to perform. So this is the context. Then the second part of the context is regarding the macrophages. And the macrophages are what is called tissue-resident macrophages, are present in every tissue. We know them with different names. In the brain, they are called microglia. In the bones are called osteoclasts. And in the liver are called Kupffer cells, but in other places are just more easy to recognize because they are called macrophages, either alveolar, splenic, intestinal, testicular, or sinusoidal, but also the heart has macrophages. And this is actually the focus of the work of Andrés Hidalgo and José Nicolás Avila, and the presence of macrophages in the heart in a normal situation, not upon an insult, was already documented. Here you see in GFP, the macrophages present. This is coming from this Pinto et al. Paper, but the real amount of these guys was not really clear. And then what Andrés was starting to do this analysis. There were some reports in which the function of the macrophages were obviously participating in immune defense, quiescence, or in maintaining the health of the cardiomyocytes, or when there is an insult, just trying to eliminate the debris of the cell's death, or help to angiogenesis, or to induce the fibrotic scars. And also, it was proposed a more specialized function in modulation the electrical conduction. Okay, but there's still unexplored the possibilities of the role of the cells in this organ. So the first question that Andrés addressed was how much of these guys are present in the cardiomyocyte. And this is a beautiful film that they do. They clarify the organ. This is a mouse heart that you see in white the vessels, and in green the macrophages that they detected and then they got a level of resolution that they allow them to really to identify, pinpoint, and count the number of macrophages that were present in an entire heart. You see now that this is this quantitative analysis that could be done, detecting the individual macrophages. So this also you can they were able to label the cardiomyocytes, and they could estimate how many macrophages were actually close by or interacting with cardiomyocytes, and it tends to be an average of 5, but also each macrophage, it was an average, a close proximity of interacting with also 5 cardiomyocytes. So the number of macrophages was quite elevated. So the herd is not only cardiomyocytes, it's other cells, vessels, but also macrophages. So what is the function of these guys, beside the ones that were already known? And for understanding that, what José Ángel did was just purify the different surface markers, different population of macrophages. They were called MAX1, Cmax, C for cardiac, MAX2, or LYSC positive cells. And then they asked what they are doing. And for understanding what they are doing, they have a mice model in which they put tomato red compounds in the cytoplasm of the cardiomyocytes, and they could really identify in situ the macrophages. Here is in blue with the nucleus, and they identify that there were red material inside the macrophages that it's very likely that are coming from a cardiomyocyte. In fact, they identified that they were red stuff significantly smaller than the macrophages that were in the outside of the cells, and some of them also inside of the macrophages. They could, so the first hypothesis is that maybe macrophages are really capturing this what was called exosphere by similitude with a previous nomenclature proposed by in the C. elegans neurological system, that they also release some things that are bigger than exosomes, significantly bigger, but smaller than cells. Then the first thing was to identify if this is actually, as the hypothesis suppose, that the macrophages release these guys, sorry, cardiomyocytes release these guys and macrophages take them. For that, José Ángel purified cardiomyocytes labeled with GFP, and he purified Cmax labeled with DS red, and he put them both in culture. And when they were in culture, he was trying to videotape if there is transfer of material in one side, in both sides, or no transfer of material. So you identify some of these intense green stuff. You see here how this vesicle, you see I'm going back, how here it appears a vesicle that moves to the macrophages, then is end up engulfed in the macrophages and then disappear. So José Ángel was able to quantify this and to monitor this, and was able to document a real transfer of several and only unidirectional from cardiomyocytes to macrophages. So yes, macrophages are able to pack, to make some vesicles that are sent out and taken by the macrophages. Okay, so then the question that Andres and José Ángel address is, so is this really significant for the function of the homeostasis of the herd, and how we can test it if this is significant by just getting rid of the macrophages? And for that they use another cell model in which they express the diphtheria toxin receptor only in the macrophages and not in the other cardiac cells, but macrophages in all the animal, and they then injected diphtheria toxin in a way that they will, when it's injected, will only kill macrophages and will not affect the life of the other cells. So this was done with a number of injections over a period of 3 weeks, and they documented that the macrophages were really down significantly and very quickly. What are the consequences of this loss? The first thing is that the exosphere released by the cardiomyocytes were accumulated in the extracellular area of the heart. But then after a while, they're no longer accumulated because the cardiomyocytes stop releasing them. And in the cardiomyocyte, what happened is that there was an induction of inflammation, probably because the accumulation of this debris outside, and also an increase in the caspase activation and the active caspase. So the cardiomyocytes are suffering because the macrophages are not there. And also they could document that there was an affectation in somehow in the autophagy within the cardiomyocytes, because the proportion of L3-2 per cardiomyocyte was really affected. And this was dependent of the activation of the inflammasome, because when you inhibit the inflammasome, you recover the normal situation. And very interesting as well, this correlates with the loss of release of exospheres, because you partially recover the exosphere release in the long term when you inhibit the inflammasome. So we will come back to this later. So So what happened? We just noticed some things in the hair, but the more important thing is that the hair function was really affected. They have affectation of the left ventricle volume and also the function of the left ventricle, and so with diastolic dysfunction. There are many other data in the function of the hair in the paper, but I just, for the sake of time, I'm gonna only focus on some aspect. One thing that it was very interesting observation is that if we analyze which is the avidity of glucose by the heart, you can do it by PET glucose with a fluorescent FDG uptake marker, and you see that when you depleted the the Cmax, you increase the avidity for glucose of the heart very significantly. And then we also noticed by 31P MRS that for maintaining the phosphocreatine levels in normal, the amount of ATP in the multiplated was significantly reduced. So the phosphocreatine ATP ratio was increased as a sign that there are these guys are probably taking glucose because they are having troubles to maintain the ATP production. Remember, ATP contribution to phosphocreatine is coming from the mitochondria or coming from the nucleus, and the mitochondria produce ATP, and the ATP goes to the intermembrane space where there is a specific creatine kinase in the mitochondria that phosphorylate, convert the ATP in phosphocreatine, and is transferred by the BDAC to the outside to travel to the sarcomere, because ATP is not going directly, it's going, the energy is going as phosphocreatine. And glycolysis can produce ATP because there is another phosphocreatine in the cytoplasm that also can do the job. So then José Ángel was able to purify these exospheres and analyze it by proteomic. This was a proteome of the whole hair, and notice that has a really indication of dysfunction of the mitochondria and a metabolic alteration. And again, with the same picture, remember that we showed the phosphocreatine was maintained, but the ATP level was reduced, and this could be really due to the fact that the phosphocreatine production by the mitochondria is impaired and has to be compensated by the phosphocreatine production by the cytoplasm, and this might be the reason because it takes too much glucose. But this was confirmed because we see that after 20 days removal of the Cmax, there is really a reduction in critical components of the phosphocreatine delivery from mitochondria, like the ANT, or like the VDAC components. Also, there are other elements in the mitochondria that are affected, like the proteostasis and several others that I don't have time to mention here right now. But more precisely, we could identify that the switch in metabolism reflected by the uptake of glucose or the phosphocreatine ATP ratio was also obtained with this Beck index, that is an index of who is producing the ATP in the cell, and the ratio between mitochondrial versus cytoplasmic. So the mitochondrial was reduced, and so the Beck index is reduced. But Ana Victoria purified the mitochondria from these animals, and tried to analyze the rate of synthesis of ATP with glutamate and malate or succinate, and was also detected that these mitochondria were really has problems to produce ATP. So there is a switch in the metabolism due to the really a loss of capacity of the mitochondria to deliver ATP in these cells. So why there is this dysfunction? One thing that it was noticed is that this in depleted C-depleted there were mitochondria in the outside the cell, close to the cardiomyocyte, that was not present in the control animal. And also, it was a surprise, the amount of mitochondria that remained in the cardiomyocyte was increased significantly. But not only despite of that, this mitochondria was less functional. So the important element that came also for the analysis of purification of these exospheres is that mitochondria were really detected inside the exospheres that are engulfed by the macrophages, and they could be done by electron microscopy, but also by immunodetection of components of the mitochondria. In white, this is TON20. There is a lot of mitochondria in the cardiomyocyte, but there is also quite a lot of mitochondria inside the exospheres. And just to show that this is also happening in humans, Andres could have access to some samples of human hairs and show also that there are exospheres containing quite a lot of mitochondria. The point is that things are starting to really meaning something. So you have exospheres in mitochondria, exospheres engulfed and that contain mitochondria, and the obvious suggestion is that mitochondria detected in white, and here the mitochondria from macrophages, and also the mitochondria from inside the exospheres. This might be really mitochondria coming from the cardiomyocyte, but something is really obvious but needs to be demonstrated. And one of the things that was done before is that was a method of differential centrifugation to enrich in a fraction containing the macrophages that could, as I told you, it could lead to the specific analysis of the protein content of these guys. And there are many information, but here is a quite interesting information. Well, first of all, there are immense amount of mitochondrial proteins, much more than in the cardiomyocyte, that are also the more abundant ones. But interesting, you can really get in red the whole herd. These are different mitochondrial proteins, and in exospheres they are increased with respect to that of the cardiomyocytes. But some of them are particularly interesting because only a few of them are not increased, like cytochrome C or AFM1, that are pro-apoptotic factor that if they are released by mitochondrial-induced apoptosis, they are reduced, and also proteins that are really participating in the structure of the membranes, FIS1 and OPA1, are significantly reduced. So those are in the exosphere. So then Jose Angel and Ana did an analysis of the different type of mitochondria within the cardiomyocyte, mitochondria that are close to the nucleus, that are presumably mitochondria that are younger, mitochondria that are already organized in the intermediofibrillar that are the more functional, and mitochondria that are in the surface, two mitochondria that are also released in the exospheres, and they qualify each organelle by a criterion of a normal aspect or obvious abnormal aspect, like broken or disorganized. And they notice that coming from here to outside, the normal mitochondria were reduced and the abnormal mitochondria were increased. So it seems that there is a gradient of loss of quality, structural quality of the mitochondria, and the mitochondria that are sent out has really mitochondria are not good. To get some functional information to confirm this impression, they perform also an analysis that is classical as assessing the membrane potential of the mitochondria. This is a fibroblast in which you can have an fluorescent that is accumulated according with the membrane potential. If you use a drug that drops the membrane potential, FSP, you see the drop in membrane potential in the fibroblast, and you can increase the membrane potential just by giving an inhibitor of the ATPase oligomycin, and then you see the response. And so these mitochondria are alive because you can modulate the membrane potential with drugs. You can do the same in with cells that contain much less mitochondria, like leukocytes, but the mitochondria from exospheres are completely insensitive either to FCCP or oligomycin, what is suggesting, as the structural indication suggests also, that those mitochondria are non-functional. And also they have, as a third proxy, a very low activity of citric synthase. So the question is, does it mean that some of the mitochondria that we expected to be not functional and therefore eliminated by autophagy, instead of eliminated by autophagy, are sent to macrophages to be degraded? And this analysis it was a good hypothesis, but this analysis needs to be really confirmation. And for that, Anna and José Ángel took advantage of a mice that was developed by Ana Latorre in our laboratory, in which we were able to generate identical nucleus, black 6 nucleus, but two different wild-type mitochondrial DNAs that can, as you see here, can be distinguished by PCR and RFLP very easily. So the experiment done was actually to use this, we named this black 6 nucleus NCB mitochondria, black 6 nucleus, black 6 mitochondria, or C57 mitochondria. So the idea is to get the donor bone marrow, to eliminate the bone marrow of our mice in which cardiomyocytes are labeled with a red fluorescent protein. And then of the bone marrow and the derived macrophages will be having the NCB mitochondrial DNA, but the cardiomyocyte mitochondria will have C57 mitochondrial DNA. The rest will be identical. And to distinguish those macrophages that actually are not killed by radiation, we have also a different marker that is a variant of the CD45 antigen, 0.1 or 0.2, that can really allow us to sort them different. So then we sorted the donor-derived macrophages to analyze which DNA they have, or the macrophages that were not killed by the irradiation. These guys, they has to have black 6 mitochondrial DNA, and this is what they show. But these guys that has to have only NCB mitochondrial DNA, you show that they have NCB and also black 6 mitochondrial DNA. So this is an evidence that the mitochondria from cardium are coming out and are taken by the macrophages. And because we eliminate macrophages everywhere, we put select macrophages in the intestine, bone marrow, spleen, liver, and we don't see any uptake of black 6 mitochondrial DNA in these tissues, but we see a little bit in the skeletal muscle. We are now exploring if there are other tissues that has this kind of transfer. but it is in heart more relevant, and also, but significantly, in the skeletal muscle. So this allow also another experiment that is interesting, which will be the fate of these macrophages. That is, the macrophages purified in that way can be maintained alive in culture after sorting. And we notice that a mitochondrial DNA external, the black 6, was really lost very quickly in hours. So these mitochondria that are coming, including the mitochondrial DNA, are degraded inside the macrophage. A second proxy to demonstrate also this transfer was done with a very interesting protein, is KEMA. KEMA was expressed, or was a vector expressing KEMA was packed in an AAV9 virus, and then the animals were infected, and you see that KEMA is red. You see that 2 weeks or 5 weeks after infection, you see the expression of the KEMA in the heart of the animals. It was a heart-specific promoter. And KEMA has another property. It's the KEMA targeted to the mitochondria, and this KEMA can change color from green to red when it's becoming more acidic. And this has been used by others to follow up mitophagy. So then the KEMA give us two types of information. You see the KEMA, we see the KEMA in the macrophages, and we also show that this KEMA in the macrophages is becoming more acidic. In the same experiment that we did with the mitochondrial DNA, we saw that the KEMA is immediately degraded in the macrophages, also indication that all the stuff that's coming from cardiomyocyte to the macrophage is degrading.
Oh no, this is just to tell you your 35 minutes you wanted me to give you.
Okay, perfect. Then the last, just to confirm this degradation, we were being able to label that the exospheres are and KEMA are really incorporated in the autophagosomes within the macrophages. So there are some calculations that allow us to estimate that there is still mitophagy, significantly high proportion of mitophagy within the cardiomyocyte, but up to 60% of the mitochondria are really degraded through this exosphere and can convert to the macrophages to be degraded. Then I really wanted to give you time for talks, and then I'm gonna go very quickly with these two questions. Is this just a basal mechanism, or is enhanced by stress? And to do the stress, what we use two models, myocardial infarction, and we see that the perinatal area of the myocardial infarction, the exosphere release is increased, and use also an overactivation, to overwork to the heart by chronic isoproterenol treatment, that we see that the damage, and we know the damaged mitochondria, and we also shows that increase the percentage of damaged mitochondria and the release of exospheres as well. And we complement this analysis with because to confirm that this because mitochondria is damaged, what is happening, we just have a model in which upon isoproterenol, we can protect the mitochondria and prevent them to be degraded. And this was actually a model developed in our lab by Enriquez by ablation of one protease, that is OPA1. And in that case, if you eliminate OPA1, you prevent the release of exospheres, because even if you have the stress work, mitochondria is not damaged. Then the second question, important, that you can read more carefully in the paper, is the formation of mechanisms of the exospheres. And I give you already one hint. The exospheres actually are released according with the expression of the activation or not of the inflammasome. The inflammasome block autophagy. If you block autophagy, you don't release exospheres. So we did several experiments activating with rapamycin the autophagy flux, and you increase the release of exospheres, or inhibition of autophagy by genetic ablation of ATG in one of the is in heterozygous, and we damage cardiomyocyte autophagy. We stop the release of exosomes. Okay. Then we also identify that the receptor MERTK in the macrophages is participate in the capacity of engulf and detect and engulf the exospheres. And if we eliminate MERTK in the macrophages, then we accumulate and we damage the capacity to eliminate. We don't eliminate completely, but we reproduce the damage also in the mitochondrial defective accumulation in the cardiomyocytes, and we also start damaging the function of the heart. I'm gonna go to the end with this summary of this new discovery, but also to mention to you that the mechanism of exosphere participate in other situations in which the function of the cardiomyocyte is really affected or compromised. There is a model that we are studying in which we generate a right ventricle damage by increasing heteroplasmia in the heart. And in that case, we see that we have accumulation of autophagy mitochondria. We don't affect the macrophage function, but we really reduce the exosphere release because this blockage in autophagy. So the autophagy connection with the exosphere function is very important. Now, you know that it's not exclusive of mitochondria. We are exploring where it's more functional, but one thing that could be very relevant is in the function of the pigment epithelial cells, retinal pigment epithelial cells. And also there are some publications that might really be relevant also in adipocytes. And with that, I would like really to thank you for your attention, and I'm open to questions if you wish. Thank you very much. I think that Andres might be also in the audience and can really participate in the discussion if he wishes. Thank you.
Thank you so much for that. It was super interesting.